Okay, thank you so much, guys, for coming. We're going to revise for midterm 3 today. I mean, it's a review. So it's going to be digestive system, urinary system, and respiratory system. It's only 3 systems, so but it's a bit of information. All right?
Really?
Really? Surprise. Okay, guys, as usual, the first thing we do is talk about exam format. I think I picked up blank. Like colors. This one is going to be slightly different in that we're going to have 4 sections in it. So section 1 is our usual multiple choice, and we're going to have 25 times 1 equals 25 points. And then section 2, this is going to be matching, since we got the digestive system, which is a progression of organs. Yeah, we kind of sort of talk about, say, the GIT, for instance, and you might have a nutrient going through the system and that doesn't get absorbed, and the structures through which it passes, it could be like blood flow in the liver and bile flow, or it could be urinary system in the nephron and stuff like that during the reproduction. It could be any one of those systems that lend themselves to putting structures in a sequence. So that's how it's going to be. So I usually just call it the matching or sequencing. It's going to have, you may have 10 things to put in order, times 1, 10 points. And then section 3, the usual short answer questions, right? And this is going to be 5. This is a 5 of 6 as usual, times 2, which is 10, 20, 45. So we need 20 points right now. Section 4 is going to be the usual long answer. And it's going to be 4, 5 times 5, which is 20, 30, 40, 65, as usual, right? So that's the exam right now. So the dimension part is actually the fun part, because usually I give you the structures to pick from. Okay? I'll give you the structures to pick from, and I'll kind of go through what things that lend themselves. So you gotta know the digestive system.
What's the long answer, 4 or 5?
What's that?
4 or 5.
4 or 5 as usual, 20 points. Okay? So you gotta know the function, which is digestion, taking in of food, right? And breakdown of food, okay, absorption, all right? Those are the basic functions, and then you excrete the waste, okay? This one includes 2 things, right? Mechanical digestion, what organ of the digestive system takes care of that? Mechanical, it's located in the mouth, but which organ? Teeth, of course, the accessory organ, and then of course, chemical, and what takes care of that? Enzymes from all over the place, okay? This is like looking at the whole system here. Amylase starts in the mouth and so forth, and then, oh my God, absorb. See what I'm saying? Absorb nutrients. So I did this at fast, like at 3 in the morning or something. Okay, and then excrete waste. Okay, so gotta know the functions of this, and of course gotta know the accessory structures and the system, the structures involved, right? So that's the basic part. Every time we have to know and then of course we've gotta know the structure of this system we're talking about. 2 groups, of course. The GIT is what lends themselves to that kind of matching kind of question, because we start in the mouth, go through the pharynx and then the esophagus and the stomach. So most of the structures you know about it, and the 2 sphincters, cardiac on top and the pyloric on the bottom. So that these are kind of freebies, because it's more like anatomy. So this should be fun questions. So any one of those systems that is a sequence of structures involved in a certain function, those are the ones I use for the second session. All right, and then the accessory organs, you just mentioned 1 teeth and all that. You gotta know that, and then the pancreas, the liver, all those produce enzymes and hormones that control digestive function. So we you're gonna know those. And then of course, you got the tunics, okay, which are the layers, okay. The adventitia on the outside is the connective tissue, all right. And then the mucosa, which is the innermost submucosa right underneath that, and then the muscularis, right? Mucosa, you know, that it's lined with epithelial cells. Some of them are, you know, goblet cells that produce mucus. Some of them are glands that produce, you know, hormones and stuff like that. Okay, and the submucosa. That's where you have the nerves, the plexuses that control the enteric nervous system. Okay, and you also have what other system do you have located in the submucosa? Starts with an L. Lymph nodes. You know, any system that takes from the outside, it always has the lymphatic system to protect, right? So you got the lymphatic system in here, okay, as well, acting up, and then of course the muscularis, which is the muscular layer, okay, and then the adventitia on the outside, sometimes called the serosum, connective tissue, right? Okay, so and based on what's located in there, there is the function of the mucosa, and you know what that is, lining, the epithelial lining, and the submucosa, we call that lymphatic system. So you gotta know those 4 layers of the GIT throughout, and then of course mucus secretions to protect and to lubricate as you go down towards the large intestine. You know that, and then stomach and small intestine, the mucosa. All right, there are all these pits, intestinal pits, gastric pits. Those pits are, you know, lined on the villi, and they contain all these glandular cells that produce something. So that pit, you know, you have like a villus like this, lined with epithelial cells, right? And then goes like that, lined with epithelial cells like that. So some of these, in these little crypts, they might be parietal cells, they might be all these kind of cells, and so forth, that produce, you know, all these enzymes and hormones. So those crypts are there in the stomach, like gastrin is also produced by some of those endocrine cells. All right, so gotta know those enzyme secreting cells and hormone secreting cells. Some of the parietal cells, like in the stomach pits, they produce what? HCl. Okay, so gotta know the cells and their functions and what they do. All right, so those are the 4 layers I just talked about. The mucosa, the epithelial lining that is some of the glandular cells. All right, in those pits, there's gastric pits in the stomach, there's intestinal pits in the small intestine, right? And then the submucosa, of course, is those lymph nodes and so forth. And the plexus is located in there, the enteric nervous system. That's why it's called the submucosal plexus. That controls secretions. And then in here you've got the myenteric plexus. That controls what? Myenteric controls? Yeah, muscles, which means?
Peristalsis.
Motility. See what I'm saying? So you gotta know what's in those structures, in those layers, and the functions of the components of that layer, right? Makes sense? Okay? So this is submucosal plexus secretions. Okay, gotta know that. And then myenteric plexus motility. Okay, that's the ENS, and then the serosa is adventitia, also connective tissue. Okay, connective tissue. All right, and then so that's the enteric nervous system there. We got the submucosa and then the myenteric. So we know what these guys do, and then regulate digestive activity in terms of motility and secretion. All right, and then types of enteric neurons. What do we got? We got motor, and we got sensory. You know, the sensory is responsible for the tactile stimulation, chemical stimulation when acid gets in there. Okay, so you got, and then motor controls the movement, okay? And then, because this is an enteric nervous system, it actually has what are called interneurons. Yes, see what I'm saying? So there's all those three types in there that kind of forms a little nervous system of its own that doesn't need control from the central nervous system, okay? But then you have other, the central nervous system, the vagal stimulation from the number 10 cranial nerve stimulates activities because that's the parasympathetic, okay? Sympathetic in this case will actually halt digestive activities, okay? Because it reduces secretion and so forth in motility. Because sympathetic is reserved for fight flight. And then of course the peritoneum, okay, which kind of covers the abdominal cavity, right? And also the pelvic cavity. So, and then we've got the mesentery, the mesentery that kind of suspends, that is attached to the body wall and suspends the small intestine, right? Okay, so includes the mesentery, okay? Mesentery with an E-R-Y, okay? And then those serous membranes, so that will open to the outside, all right? And then the serous membranes. So you can, and then of course, you can guess the and then of course some of these things, like the oral cavity, I just want you to know what function goes in there and just the basic, structures in there. The mouth, the vestibule is between the lips and the cheeks and the teeth. See that? When I put my tongue in, that's the vestibule. And the oral cavity proper, of course, is from posterior to the teeth and in front of the pharynx, right? Okay. And when your mouth is closed, the oral cavity is completely filled by your tongue. Yeah, the tongue completely fills it up when your mouth is closed. Okay, so know the parts there, and of course the cheeks, and it's stratified epithelial cells that prevent abrasion, of course, just like skin is stratified epithelium. Okay, and then the palate, of course, it's got the two hard and soft, and the functions of both. The soft palate makes up the uvula, which closes up the posterior nares, or the chewani, if you like, when you're swallowing, so that food doesn't get into your nasal cavity, right? And the hard palate, what's the function of the hard palate? Where is it involved in?
Swallowing.
Yeah, swallowing. When you push up with your mouth closed, your bolus of food up against the hard palate, it will go to the back. Okay? Try doing it with your mouth closed and it will just ooze out. Okay? But if you do it with your mouth closed, it's got nowhere to go but back. Okay? So, accessory glands that are located in the head, really, what are they? What are the accessory glands in this region of the body?
Tonsils?
Yeah, you got tonsils, on the throat, but, salivary glands, okay? The tonsils come a little after the oral cavity because they're in the pharynx, okay? But salivary glands, of which there are three, right? The parotid, which is kind of right here, and then the submandibular, which is here, and then the sublingual. And I want you to know the functions of those, okay? The parotid mostly enzymatic. It's a serous fluid, watery fluid, with enzymes such as this enzyme, really, salivary amylase. The serous one is also, submandibular is also kind of serous fluid with a bit of mucus.
The sour one?
This
Saliva when you get something sour.
Yeah, when you get something sour or something really nice, you sometimes, even I get some tingling feeling around here when I'm eating something I really like. Okay, so that's the parotid, which really has the salivary amylase and the watery one. And the sublingual is mostly mucus. So the sublingual one, the function, the one that's under the tongue, is to lubricate the voice. See what I'm saying? Okay. So gotta know those three. And then of course the tongue, we know it's a muscle, parotid, enzyme. So it's a serous fluid, okay? And then submandibular, under the mandible, okay? And that's serous and mucus mixed, okay? And then of course, sublingual, under the tongue. That's mostly mucus for lubrication, Okay, so sometimes I ask about the and then of course teeth, mechanical congestion, you know that. Okay, good. So,
So we're going to need to know anything about the teeth?
Not really.
Oh.
I just wanted to know that they're involved in
Crown,
mechanical
back,
digestion, and just know the basic structure of it. Enamel on the outside, dentin in the middle, okay, and then the pulp containing the nerves and blood vessels, you know, right there in the middle, and then you get the cement and the, you know, those ligaments that attach the teeth in the roots. But just the basic function of them is all that I really need you to know about that. Pharynx, of course, the throat, you know, is involved in swallowing. All right. The esophagus conducts the food down via peristalsis. And then you got to know the swallowing mechanism, right? So you got the voluntary phase where you push up your food, your bolus against the hard palate. Once the bolus of food gets into the throat area, into the pharynx, it becomes reflex. Okay, you have those esophageal muscles. I mean the you know the constrictor muscles, upper esophageal constrictor muscles relax, and then there's a wave of this contraction of starting with the superior one, going to the medial one, in the lower one. They contract sequentially to push the bolus of food down in the pharyngeal muscles, right? The constrictor ones will relax, so that in the upper esophageal constrictor muscles. And so this is, you know, reflex. And then, of course, once you get here, you get the involvement of the vocal cords and vocal folds. Okay, that will also close up the glottis, and the epiglottis closes up that during the reflex pharyngeal phase. And then, of course, because sometimes I ask about the swallowing mechanism and for you to explain it. And then once the bolus of food gets into the esophagus, another reflex action is set up, and then you have peristalsis, okay, which is a wave of contraction of the circular muscles. Peristalsis is, okay, behind the bolus, and you know, relaxation in front of it. Make sense? So you kind of sort of squeeze it through this, and you open up the front for it to go. So that wave just goes like that. Contraction towards the back of it, and then in front of it, relaxation. So that's what pushes it down. So that's the first point question I usually ask about this phase of relax. And then, of course, peristalsis, you know, circular muscles involved, and you get a wave of contraction behind the bolus, and in front is a relaxation. Right? Okay. The stomach, of course, we know the function of it. All right, is to convert bolus into what? What does the stomach do? It convert the bolus of food that you just swallowed into. Starts with a C, right?
Chyme.
Chyme, yes. And what is chyme?
It's a mixture of the enzymes.
Bolus and the gastric secretions. Okay, that's what it is. That's what it is. So you know the functions already, and you know the characteristics and the regions of it already from the anatomy. Okay, and you know the food loose and so forth, caloric region and whatnot. The sphincters, you already know that, it was in the exam. The cardiac up there and the caloric down there. The regions, you know, so we're not going to worry about that. And then, of course, the stomach wall is basically the same, kind of 4 layers. The external, which is connective, and of course the muscular layer. In fact, there are 3 layers of muscles in here, in the stomach. Why? Because it controls in all sorts of directions. So you get oblique muscles, you get circular muscles, you get longitudinal muscles, you get all that stuff. Because it's a bag, a irregularly shaped bag, that has to do all that mixing. Okay? And so forth. And then, of course, the submucosa, you got your usual lipoids, and so forth. And then simple columnar epithelium. Because some of the cells will produce, some of them will be glandular, some of them will produce hydrochloric acid, right, of cells, right? Okay, so those are them. In the gastric pits, it simply means, you know, around the villus like this, then you got the microvilli, right, or whatever it is, or circular folds, and then you got these cells here, lining them. So these are the pits where some of these cells, the surface mucus cells will be on the surface of the pits. So that will be towards here, you know what I'm saying? And then the mucus ones, you know, the neck, you see the pit is like a long tube, tunnel tube. So like the neck will be kind of somewhere here. You get more of those mucus cells because those ones have to be on the surface of because the mucus is going to be produced and stays on the surface of that organ so that it doesn't get digested, right? With the enzymes, number 1 and 2, with the acids too in the stomach. Because the stomach is producing a whole bunch of hydrochloric acid from the parietal cells. Okay? What do the chief cells produce? Enzymes, right? Pepsinogen and so forth. All these enzymes are produced in active form, right? Okay? And then the endocrine cells, what do they produce? Gastrin, hormone. Gastrin, guys, you know, in the stomach, which causes contraction of the stomach and so forth, and also produces histamine. Okay, histamine is kind of brings a bunch of blood to the digestive system. Okay, because histamine is normally an inflammatory agent, right? Okay, so gotta know all these cells lining the gastric pits and what they're doing, right? Most of the time, the name tells you what it's doing, and where it is too. Okay, gastric juice will contain all these things that have been secreted by these guys, right? From the hydrochloric acid and you know, the enzymes, you know, and so forth. Okay, and the hormones that go into blood. Okay, so the gastric juice, and then you got that proton pump, right? Which is in the which of these cells contains this? Which of those cells above? I just told you the one that produces the acid, right?
Parietal.
Okay, see what I'm saying? Because then this exchanges the H plus and K plus to maintain electrical neutrality, right? The proton pump in the parietal cells, the H comes into the stomach, and then the chloride will come in as well, okay? And then we form hydrochloric acid, okay? And the chloride is opposed by the bicarbonate ions and so forth. Because bicarbonate ions are easily available in the blood. Why? Why are they easily available in the blood? Bicarbonate ions. Because that's how carbon dioxide is transported, right? We get H2O plus CO2 equals H2CO3, which is carbonic acid, right? Which splits into HCO3 minus. So that gets exchanged, so you maintain electrical neutrality. You get your H, you get your Cl, sodium chloride remains salt in blood, the sodium all over the chloride all over the place in the blood. Okay, so you can make acid very easy. And hydrochloric acid is a very powerful acid, by the way. Okay, so regulation of stomach secretions, of course, the gastrin, of course, which is a hormone that causes increases secretion. Pretty much when the stomach distends, okay, it will secrete that hormone. Those cells in the neck, and then come back and act on these cells. Because gastrin is just released into blood, comes back and acts on these guys. All right, endocrine cells actually, only, and then produce the gastrin, and then these guys will start to produce the enzymes, the pepsinogen. Parietal start to produce the acids, and then you get all that gastric juice. And then, of course, movements, mixing to form the chyme, okay? And then, of course, the peristaltic movements to empty, okay? All right, so very good. Now we go to number 2, digestive system 2, and this we're talking about the lower GIT and finishing up the digestive activities, right? So we're talking small intestine, and the small intestine is very long, made up of the ileum, which is the longest, right? About 12 feet long, and then the jejunum, about 8 feet long, and then the foot-long duodenum. So we know about all those divisions, right? And then we talked about how it's got the small intestine, it's got folds like this, and then it's got villus, Okay. And then we talked about how it's got microvilli. And we said it ratchets the area to the tune of how many? How many times? Right? 600 times. Increases area. And that's because of that. Okay? And then, of course, the intestinal glands in the pits, all right, that will produce all these peptidases, all right? Some of them will produce cholecystokinin, right? Some of them secretin. In fact, as soon as the chyme from the stomach enters the duodenum, you release secretin. Secretin is what causes the pancreas and also the gallbladder to release a watery fluid containing bicarbonate ions, alkaline solution, to neutralize the acid. Otherwise, it will go right through the small intestine, which is, because the pH is about 1-ish, 1 point something, that's as acidic as you can get. All right, small intestine epithelium inside, small intestine epithelium. You get all those cell types, right? So you got the absorptive cells. What else you got? Goblet cells. What else you got? Granular cells, which are also called, what are they called? Yes, Paneth, that's good. Steph, Paneth cells. And what do they do? What are these Paneth cells? What do they do?
They're macrophages.
Yeah, they're macrophages of the small intestine. And then the last type is endocrine cells, right? Because you got to produce those hormones. What are those hormones that I say are produced by the small intestine? 2.
Carcinogen and gastrin? No.
Are hormones. 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 Gastrin is the hormone in the stomach, right? But the hormone in the small intestine would be, there are 2 of them. I just mentioned them when I started. Secretin and the one who starts with a C, cholecystokinin, okay? All right, and of course you got the pious fetches, which is lymphatic nodules. Okay, you know what they do. Of course, it's involved in getting all the bacteria and whatnot, including the granular cells that have to do that. Okay.
So the hormones in the small intestine are secretin and what?
Cholecystokinin. Okay, secretin causes the secretion of alkaline fluid for the most part. Okay. And then from both the pancreas and the gallbladder, okay? And then cholecystokinin causes the enzymatic secretion from the pancreas, from those acinar cells, right? Cholecystokinin also causes bile release from the gallbladder to the bile salts for fat digestion. You see what I'm saying? Okay, so I want you to know those 2 functions of secreting, the 2 functions of cholecystokinin. Okay, these are intestinal hormones. All right. Okay, so gotta know, of course, the cell types, because the cell types will tell you the function of that particular part of the GIT, right? Because it will, they will produce something or do something which is involved with the function of that particular, part of the small intestine. You know that the small intestine is the major digestive organ, right? Why? Because it does 2 things, which is absorption and finishes the enzymatic breakdown. Okay, because the stomach only breaks down amino acids to peptides, which are small little chains. Okay, we need to go all the way to amino acids. So the small intestine finishes that up, and then the amylase and salivary and in the stomach only splits the carbohydrates into disaccharides. Okay, you need to go to monosaccharides. Okay, and that happens in the small intestine. Of course, you gotta have these secretions. All right. Okay, mucus to protect immediately from the acidic chyme. All right, and then this is stimulated by these things. Okay, the chemicals, for instance. Of course, the acid is gonna really cause a whole bunch of activity in there, because you don't want the acid to digest through the small intestinal wall. Tactile, which is touch, mechanical receptors. So you get chemical receptors, you get touch receptors, which are mechanical receptors, okay, up in there. And then, of course, vagus stimulation, which is cranial nerve number 10, stimulates digestive activity, right? Vagal is the parasympathetic branch of it, stimulates digestive activity, and then secretin, of course, will cause the pancreas to secrete a watery fluid containing bicarbonate ions, which is alkaline. And then it will cause the gallbladder to secrete a watery solution containing bicarbonate ions as well, which is alkaline. To get rid of that acid, because that acid is like, it's very acidic. Small intestine, of course, is get the segmental. Remember the segmental? What is the function of segmental contractions? What is the function of segmental contractions in the small intestine?
Mass movement.
No, that would be peristalsis. So segmental would be the only other part. What does the what do you have to do whenever you get to a part of the digestive system? You gotta mix whatever is coming in the food with the secretions. You have to do that. You've got to have mixing movements. So the segmental movements will be the mixing movements in the. Remember, I told you how the circular muscles are involved in segments, and they contract like that, and then it will switch the contents around like this, and then they finally mix. Peristalsis is moving material along. Okay, so you gotta know these things, guys. Okay, if I had to ask you to talk about the functions, the movement or motility in the small intestine, I expect you to tell me about what it is and tell me what the function of that motility is. Okay? Contraction stimulated, of course, by distension. Whenever you get contents in there, you stretch this wall of that organ, and then, of course, local reflexes. That's why you have the enteric nervous system, which is a kind of small nervous system all by itself. Okay, that doesn't necessarily need a lot of CNS control, except through the vagus, which kind of comes from cranial nerve number 10. Okay, but for the most part, it's doing it by itself. Okay, so you have a lot of local reflexes, and of course the peripheral nervous system, which includes the vagus nerve and the parasympathetic nervous system, actually. Parasympathetic. Okay. All right. Very good. So here we go. Now about the liver, I just want you to know the basic structure of that. We got the 4 lobes of the liver in the human. Okay. Quadrate, if you will. Quadrate, right and left median lobes. That's all. And then those are separated into 8 segments. Right? Then the 8 segments have got lobules in them, which are hexagonal, like so. All right, and then we got, you know what I'm saying? We got the duct there. We got that, and the portal vein in there. You got on the triad, the portal triad. So you gotta know that. You gotta know what the hepatic duct is. The green that carries bile away from that, and then you've got the columns. Okay. The columns of tissue. In the center, you've got the vein, okay, the central vein, and all these guys live in there. But you've got cords or columns that radiate outside, okay, of those hexagonal hepatocytes, right? And in between them, you got the bile canaliculae and so forth that join up to form the hepatic duct, which goes down with those ducts that we did in the lab, so we know all about that, and it ends up in the common bioduct, right? And then you got the hepatic portal branch on that portal triad going in together with the hepatic artery branch, and they form the hepatic artery branch and the hepatic portal vein branch. They join to form what? Which goes all the way to the central vein. The sinusoids, those big old kind of like blood vessels, okay? Sinusoids that are lined with all these Kupffer cells in it, which are for, what are Kupffer cells for in the liver? What about granular cells in the small intestine? What are they for? Yeah, those are the macrophages of the liver, right? So I want you to know these cells, their functions. Okay, guys, this is kind of like, it's a little much, but you know, I want you to just kind of think about what you are trying to do with the digestive system, the purpose of it. Okay, and you know, anything that opens to the outside, guys, always has to be guarded by these immune cells. Okay, because when you open from the outside, you get bacteria coming in, so you gotta take care of that. All right, and then the sinusoids will. This is another thing that might lend itself to those matching stuff. You know, I could say, okay, line up, you know, blood flow through the liver, you know, as it comes, you know, the nutrient-rich, oxygen-poor blood through the portal vein, hepatic portal vein, and then you get the oxygen-rich blood coming in through the hepatic artery branch on the portal triad. So they mix up to form the sinusoids, which join the central vein, and then the central vein, they join together to form the renal vein. Renal vein, they join together to form the hepatic vein, hepatic vein. Okay, the hepatic vein then joins the inferior vena cava, right? Going to the heart. So just know, the cords are simply columns of cells, hepatocytes, that sort of radiate from this, and then within it you have all these biocanaliculi and so forth, you know, going to the thingy like that. Okay, and then you have the arteries coming like that, and then you have the hepatic portal vein coming in like that, and these guys join to form the sinusoids, which then jump into the central vein, right? Which then becomes the hepatic vein and so forth. So, you know the function of the Kupffer cells, what are the immune system cells, apart from the lymph nodules, bias patches and stuff like that. Okay, so the liver does that, produces bile, and then the gallbladder merely stores it, but it's produced in the in the liver, and then nutrients. Okay, it deals with nutrients. It makes new molecules. All the glycogen is stored up in there. When glucagon is released, it converts it into glucose. Okay, and then it can convert glucose to glycogen. It converts amino acids to proteins to glucose. You can do that. Lipids also can be converted to glucose because that's what the body uses a lot of. Or you can replace the R group on an amino acid, okay, and form a new amino acid altogether. Because your body may need these amino acids in certain ratios that you may not necessarily ingest them in those ratios, okay? So the liver can do that, can change it, and of course detoxification. If you eat something, you know, whatever, so it does that. And then phagocytes in there, macrophages, all right, that will digest what? Red blood cells, right? Because the liver does that. They will digest bacteria, right? What else will they digest? Cellular debris, yes. See what I'm saying? So they do that. So gotta know these functions, and then of course produce blood proteins, such as albumin, things like that. Globulins, which are involved in immunity, all that. So gotta know that, all right? The functions of these organs and systems you have to know, right? And then, of course, blood flow through the liver. That's another thing that could be kind of matching thing. You know what I'm saying? For the middle part. So whichever I choose, you know, I could choose any one of these things, or urinary system, or whatnot. Okay. So of course, bowel transport. Okay. And you know all about the left and right hepatic ducts. And then the common hepatic duct, then joins the cystic, which comes from the bowel, gallbladder, and things like that. So bile, you need to know the characteristics, right? This yellowish, greenish fluid that contains all these phospholipids, neutral fats, cholesterol, bile salts, very important, because bile salts have functions of their own. What is the function of bile salts? Emulsification of fat, right? So gotta know those components of that and the characteristics, okay? And we know about the enterohepatic circulation. Huh? What does the enterohepatic circulation do? What does it achieve? What's the purpose of it? The enterohepatic circulation. It recycles bile, right? 90% of the bile is recycled, it's reabsorbed, okay? Also, bile salts stimulate more secretion of bile salts, so they go back into the gallbladder, cause more secretion, and it kind of circulates like that. Because it will be energy demanding if you just secrete all those bile salts and then just void them through the intestine, so you don't do that, okay? But you know the pigments that are in there, right? From the breakdown of red blood cells, right? So you form bilirubin, which is converted to biliverdin, and some of these derivatives, once it gets into the small intestine, it becomes urobilinogen, which then goes into the feces. You know what color that gives. Goes into the kidneys. You know what color that gives to urine. Okay, so you know all about that, and then of course regulation of bile secretion. Okay, so we get the secretin and also cholecystokinin. They stimulate the bile, the gallbladder to release different things. The secretin will cause it to produce the serous watery fluid containing bicarbonate ions, right? The cholecystokinin will cause the gallbladder to release bile salts in there. They are normally stimulated by fats, okay? If you eat a burger from McDonald's or whatever, it's got a bunch of fat in it, so you will definitely secrete cholecystokinin so that it will cause bile salts. So that's why when you get gallstones, you don't, they tell you not to eat a bunch of fat, because then you're not having enough of the bile to go and emulsify the fats. It will just cause all sorts of problems in your digestion, alright? So gotta know all that. And then of course the pancreas are mostly interested in all the functions of it actually. The exocrine function is important. What does the exocrine function produce? It produces the enzymes, right? Okay, guys? Okay, and what about the endocrine? That's obvious, right? The endocrine function of it produces the hormones. And we know what hormones that is, right? What are the two hormones produced by the pancreas?
Insulin
And glucagon, right? Okay, you know the location of the pancreas, don't you? Kind of behind the stomach and crossing across. You've seen it before many times in the cat, okay? And then the pancreatic secretions, that's that watery fluid which is alkaline containing bicarbonate ions, all right? That is stimulated by secretin, okay? And then cholecystokinin stimulates the enzymatic function. Cholecystokinin causes the release of the enzymes in the pancreas. Okay, so it stimulates the exocrine function. Okay, so gotta know that. And then of course regulation by the hormones, which we said, cholecystokinin, secretin, they cause different kinds of secretions from the pancreas. And then of course the parasympathetic, as usual, always stimulates pancreatic secretions. It causes the pancreas to contract, and then the sympathetic will actually reduce. Sympathetic system is normally for fight, flight. It normally reduces digestive activity, right? So it will reduce secretions from the small intestine, from the pancreas. Large intestine, of course, it's got the 3 divisions. We know about that, all right? Well, not the 3 there's more than just the transverse, descending, ascending colon, all right? There's also the sigmoid, and then some other parts, like the cecum is part of the large intestine, all right? It's not only just the colon, the vermiform appendix, which is the worm-like. Okay, the appendix is part of the colon, part of the large intestine, sorry. So you got the colon, the appendix, the cecum, all that. is part of the large intestine, right? And then we talked about, so we know about what are the three unique features of the large intestine in the human. We got the host, right? Which are those pockets that are created by the tension in the tenii coli. Okay, tenii coli is three bands of elastic connective tissue that have a tension in them that sort of pulls the large intestine into pockets, which makes that rostral appearance. You know what I'm saying? Like this. You got three bands of connective tissue that contract a little bit, and then they create those bags. Okay, and then what is the third feature of this? I normally like to ask this and say, okay, can you tell me about the unique feature? Crypts is not, but they do have crypts though. That produce mucus and whatnot. It's the epiploic appendages. Epiploic. Okay. I normally like to ask this question about this. The epiploic appendages don't have a function. They're just like fat, pockets lined by connective tissue. The function of which is not quite known, but they are there. All right. You know the functions of the large intestine, right? What is the function? Yeah, water absorption is the main function of that, and then of course with the excretion of waste, right? Those are two main functions, right? And then we know about the bacteria, right, which is in there. So the bacteria really has got a whole bunch of functions. What are some of the functions of the bacteria up in here? In the large intestine, in the colon.
Can you break down food?
Break down cellulose. All the plant material that we can't digest. So it's a symbiotic relationship. It's a mutually beneficial symbiotic relationship. Because not only do they get these nutrients that we can't use anyway, they also produce for us vitamin K, okay, which is good. But they have some other side effects like produce some flatulence, yeah, which is the gas, from their digestive activities, okay, and they form about 30% of the dry weight of feces. That's a lot of bugs, if you know what I'm saying. Dry weight, no water, and 30% of it is all bugs. Okay, so those are some of the characteristics and so forth. Okay, so gotta know that, and then of course, movement. You got major peristaltic movements, which are our movements, which occur a few times a day. Okay, and then you get the mucus secretion, of course. Now the third one is the respiratory system. Okay, the respiratory system. You gotta know the process of respiration, right? Which includes those 5 things, right? Like ventilation. What else? Exchange of gases, blood, and alveoli, or lungs, all right? And then you've got to transport it. Sometimes I might say, what are the processes that are involved in respiration? Okay, what processes? And then you describe them and you talk about it, you know what I'm saying? And then once you transport it, it goes to the tissues, right? So you've got exchange of gases between blood and cells or tissues, body cells, right? And then the last bit, which is, what do the cells do? Use of oxygen to do what? To create energy, right? Oxidative phosphorylation, so forth. The electron transport chain. Huh? What's that? Okay. Oh, that's use of oxygen to create energy.
Okay.
Yeah. I'm kind of sort of talking while I'm writing so that we can get a move on. Okay. And these are right there in your notes, right? If you look at that, you see that. And then, of course, it's split into 2, right? We've got the upper respiratory system, which includes the nose, the nasal cavity, the pharynx, the throat, and so forth. Okay. And then larynx. And then the lower, which is the trachea and all that stuff. Okay? And which includes the tracheobronchial tree, which is like an inverted tree. So you know those parts, you know the nose really. I want you to know about just a few of these features, like the nasal cavity with the nasal concha. And the functions of the nasal cavity really is to do what?
Warm the blood.
Warm the air. Yeah, comes from the blood, but it's warm air. Moisturize it and filter it. You know what I'm saying? You gotta filter it from particulate matter so that it doesn't go down there. Okay, that's why you've got the mucus and the pseudostratified epithelial cells ciliated, right? So you can move mucus and particles up, and then you can use the spirit or swallow it, whatever you like. Okay, so gotta know the functions of the nasal cavity and just the basic structures. You get the nostrils up in the front and the jawline in the back, which are also called the posterior nares. Okay, and then of course the pharynx. It's got those three regions: the oropharynx, the laryngopharynx, and the nasopharynx. Okay, just know where that's at. The nasopharynx is up on the top, by the soft palate. The laryngopharynx actually starts from the epiglottis and goes up in the esophagus. Okay, it doesn't go to the trachea. So it carries air and food and things like that. Okay, so and then the larynx, of course, that's where the vocal cords are located. Okay, you got the vocal folds or false vocal cords, and then you got the true vocal cords. Okay, okay, so the false, and then of course they're involved in sound production, and also the larynx kind of acts as a switching mechanism that conducts air to the right channel, food and drink to the right channel. Okay. Aided by the epiglottis and the vocal cords that shut down the trachea, the glottis, which is the opening of the trachea. Okay, and then the characteristics of the vocal cords, of course, the false vocal cords are superior, they're on the top and they kind of go down like that, and then they suspend the vocal cords on the lower or on the inferior side, right across the glottis, right? And the vocal cords, true vocal cords, are attached to those arytenoid cartilages that go like this to put tension on there. Okay. Sound production, I want you to know about the pitch. How do you create the pitch? Okay, which is the frequency. All right, and that's the tension that's created by the arytenoid cartilage, right? And then loudness. How do you create loudness? Right? Well, what creates loudness?
Increased air.
Yeah, the amplitude of the waves, which is how strongly the true vocal cords are vibrating, and that's the force of air going through, up and down. So that's what creates voice, loudness. Okay, and then things like the quality of voice, timbre, and stuff like that is resonance in the sinuses, the throat, and so forth. All right, so you just gotta know the basic sound production. The trachea, of course, it's got those 20 or so C-shaped rings that are made of cartilage, hyaline cartilage. All right, and of course it's lined by those pseudostratified ciliated columnar epithelium, right? Because then it can carry all the mucus and particles up, right? And then the bronchi, of course. I want you to kind of know how the structure changes and the function from the trachea with those cartilaginous rings all the way to the respiratory bronchioles, which are mostly smooth muscle. All right. So you start with the ciliated columnar epithelium, all right, and then by the time you end up in the alveolar ducts and so forth, and the alveolar sacs, you've got squamous epithelial cells, all right? Then you start to talk about, so I want you to know those characteristics as they go up and down. And then of course the carina, what is the carina? This is the cartilage, separates the primary and the bronchi. Yes, we're doing. What did we say about it? What's the significance of it? It's very sensitive, right? Because that's what guards, the last guard of any particulate matter going through to the lungs. So it's very, very sensitive and it stimulates very, very powerful coughing reflex. Okay?
So when you inhale something you're not supposed to inhale, like
Yeah, it will end up
with
there.
drinking, that's what's making you cough.
It makes you cough a lot. If whatever you've ingested, whatever you've taken into the trachea, if it gets there, then you really cough pretty bad. You know.
Choke, wouldn't you?
Yeah, you would, and you would really cough badly. To try and expel it before it goes down further, because if it goes further, then the tubes are getting smaller and smaller and smaller, and that's not good. Okay, and then the right one, the right bronchi is fatter and it's more vertical actually, because the right side, and this is the costal canal, and the left is kind of more horizontal and it's thinner, and then the carina will be here, and then there is the trachea right there. Okay. So the left, because of the location of the heart, it's kind of sort of goes like that. And this is more vertical. On the right, it's wider. So that's where things mostly tend to get stuck in. And the carina will kind of try to prevent things from going down there. Okay. And then, of course, the characteristics of the lung, they go down to the fifth intercostal space. They end up by the diaphragm, and they go up, about 2.5 or so centimeters above the clavicles, and so you just kind of know that. You know how many lobes on the right? 3 and 2. Okay, we got that. So you just the tracheobronchial tree and how it's kind of splits up into the primary, the lobar, the segmental, and the bronchioles, the respiratory, terminal bronchioles, respiratory bronchioles, and so forth, and how it's divided up, and then alveolar ducts and steps, and then structural, the pneumocytes, step 1 pneumocytes are the squamous flat cells that for gas exchange, of which there's 90% of these are those cells, and then these are the type 2 pneumocytes, which is the tube cells that produce surfactant. All right, the respiratory membrane, you know the components, right, like surfactant, and then the epithelia, the basement membrane of the alveoli, the epithelia, basement membrane of the blood vessel, the capillary, and then the space between the two. Those are the 6. Sometimes they ask you to list the components of that, and then the pleural, of course, the one on the outside, visceral, on the inside, and then you get the pleural cavity, which creates negative pressure with the fluid in there, and how the pleural fluid helps to keep them together, which creates that suction that keeps the lungs inflated. All right. So all those things. Okay. Respiratory system too. I'm kind of going to have to sort of skim through this kind of quickly. So just the 2 circulations in there, right? One is coming with deoxygenated blood to pick up oxygen. That's the pulmonary circulation, which we've already done many times with the heart. But then there's the bronchial circulation, which actually carries blood to the lung tissue itself. Okay. And then, of course, inspiration, not the muscles involved, like the external intercostal, the diaphragm, and things like that. Okay, sternocleidomastoid muscle, the scalene muscle, all those know that. And expiration, internal intercostals, for the most part, expiration is the passive movement by recoil of the digestive organs and the relaxation of the diaphragm and things like that. But when you have maximum expiration, that's when you have the pectoralis minor, and you have the abdominal, not pectoralis minor. Pectoralis minor is inspiratory muscle, but you have the abdominal muscles, all right, that are involved in there, okay, and then the external intercostals for expiration. So know those muscles, and then airflow, Boyle's flow, okay, pressure is inversely proportional to volume. Poisson's law just says the resistance is proportional to the diameter to the power 4. Okay? So that small changes in diameter can mean large changes in resistance to airflow, right? Okay, so that's that. You know, recoil of the lungs is because of the elasticity of the lungs, right? Of course, and the negative pressure in the pleural cavity tends to cause the lungs to recoil, right? Surface tension, because of the attraction of the motor molecules, causes them to recoil too. Okay, and then surfactant prevents lung collapse. It's got lipoproteins in it, and then it's simply a matter of wedging some other molecules between the water molecules so that the attraction between the water molecules is not that much, and you can release by tenfold the surface tension, and then the pleural pressure, of course, which can be changed by removing pleural fluid, and adding pleural fluid. All right. Surfactant, which is a surface acting agent, literally, and then it has those lipoproteins in it that kind of sort of wedge themselves between water molecules to prevent the attraction. And then you get the pleural pressure. The fluid, when you remove the fluid, you create more negative pressure so that you inflate the lungs more, you create that suction. And then when you put more pleural fluid, it creates a kind of positive pressure, all right, and so forth, all right? Okay, so measurements, you know, I need you to know these measurements like compliance, measuring lung volume, okay, and the uses of it, compliance, like when you've got emphysema, your compliance is higher than normal. It means the air is just sort of going through, okay, it's not really staying resident. And if you've got lots of connective tissue in your thing, there's more resistance, and your compliance is lower than normal. So lung volumes and capacities, you got the tidal volume, which is the lung, you know, the normal breath, residual volume, which is always stays in the lungs. And you should look at that graph for this parametric graph. Inspiratory reserve volume is what you can inspire over and above tidal, and then expiratory is what you can expire over and above tidal volume, I mean. And then minute ventilation, of course, is simply the amount of air coming into the lungs in a minute. Okay, so it's tidal volume times the respiratory rate, which is almost 12 breaths per minute in a resting individual. And then alveolar ventilation, you start to account for dead space, okay, which is about 150 mils. And all the you know, air in the non-exchange parts, like the trachea, the bronchioles, and things like that. Okay, only air in the alveolar is exchanged. So when you account for that, that's alveolar ventilation. Okay, so just know those definitions for those. Okay, Dalton's law is about the partial pressures. When you mix gases in a container, they don't become one gas. They stay. Oxygen stays oxygen, and then the volume of that gas, okay, will be the partial pressure of that gas. Okay, so the partial pressure is directly proportional to the volume of that gas in that container or mixture of the gases. So just be able to explain Dalton's law, Boyle's law, Poisson's law. Okay, and then of course the exchange of gases in the lung, oxygen dissociation, hemoglobin curve. It shifts to the left so that the hemoglobin takes in more oxygen. Okay, because there's more oxygen than carbon dioxide, and then in the tissue the opposite happens. It shifts to the right, and that's also called Bow effect. When you've got low pH, okay, oxygen is readily given off by hemoglobin. Okay, and then when that happens, you know, because in cells you've got a bunch of carbon dioxide moves into the red blood cells, and it forms carbonic acid. What happens? Then the HCO3 molecules, the bicarbonate, have to come out. So when they come out, because they're carrying a negative charge out of those red blood cells, chloride moves in. So that's the chloride shift. Okay? Remember, because most of the carbon dioxide is transported as bicarbonate ions. Okay? So the Bow effect is the movement of the oxygen hemoglobin dissociation curve to the right, which means that hemoglobin will give off more oxygen because it's in an acidic environment. It's where oxygen is. And then the Haldane effect, when hemoglobin that is given off oxygen readily attaches to carbon dioxide on the amino group, okay? And that's the Haldane effect, okay? Because in the tissues, the hemoglobin is busy giving off the oxygen for the tissues, right? And the tissues are busy giving off the carbon dioxide, right? So oxygen that is, once it comes off of hemoglobin, it will readily attach to carbon dioxide. That's the Haldane effect, okay? So that you can get rid of carbon dioxide. And then respiratory structures in the brainstem, of course, you've got the dorsal ventral respiratory groups and the pontine group that are involved with respiratory rates and managing expiration and inspiration. Okay, so those are things that you need to know. Urinary system, pretty much know the functions of the kidneys, right? Which includes excretion of metabolic waste and so forth. System functions simply mean that the kidney produces urine, the urine just conducts the urine to the bladder stores it, the urethra, so those are the functions of the system as a whole. But most of the major functions of the urinary system is the kidneys, and you know that ions, all these pH and stuff. So gotta know that. Of course, that goes without saying. And then, of course, pretty much just know the structure. The cortex on the outside, the medulla on the inside. The medulla has got those pyramids, right? And then the minor calyxes and the major calyxes, and that kind of conduct towards the sinuses and then towards the renal pelvis. Those structures kind of just know that. This is another thing that could give matching, when I could do matching when you're talking about parts of the, even the nephron, for instance, how we have the glomerulus and so forth, and the Bowman's capsule, proximal, distal convoluted tubules, loop of Henle, all those structures, when the urine is being formed, and then of course the side. So this is from outside to inside. The sinuses are the innermost, right? And then it leads to the hilum. So know the cortical nephrons, which are mostly in the cortex, and the juxtamedullary nephrons. This forms about 85% of all the nephrons, cortical nephrons. Juxtamedullary nephrons, they're next to the medulla. They've got long loops of Henle and involved in concentrated urine production. And then the filtration membrane, right? It's got to go through the epithelia of the blood vessel, the glomerulus, and then the podocytes on the visceral layer of the Bowman's capsule. And then, okay, so gotta know those components of the respiratory membrane. Okay, and then good. So the capillary beds. You got the glomerulus, the duct of capillaries in the Bowman's capsule. Then you got the peritubular capillaries surrounding the tubules, and then the loop of Henle is surrounded by vasa recta. Okay, so just know those, and then the blood flow in the kidney again. That's another kind of sort of matching where you're coming from the renal artery, which then branches up, forms the arterioles, afferent arteriole, glomerulus, then it goes into the efferent arteriole, the peritubular capillaries, goes into the vasa recta, the vasa recta goes into the veins, the branches of veins, and then which goes into the renal vein. And then that goes out. Okay? Renal vein then goes into the inferior vena cava and whatnot. And then we know that juxtaglomerular apparatus know which cells form it, granular cells, or sometimes called juxtaglomerular cells, on the afferent arteriole, and then the macular denser cells on the distal convoluted tubule. And these guys form renin, which is involved in the renin-angiotensin system. Okay, sorry about that, guys. And then, of course, urine production. Those three processes: filtration, and then reabsorption, and tubular reabsorption and tubular secretion. Gotta know those. Okay, all right. And then transepithelial just means you've got to go through the epithelia of the renal tubule and also go to the epithelia of the blood vessel for things to be reabsorbed, right? And then paracellular, it means it goes between the cells of the tubular cells, like ions and all those. They don't even have transport systems. So all the transport mechanisms that we did in ANP1, like antipodes, sympads, facilitated diffusion, are all involved in the reabsorption, right? And active transport, because the sodium-potassium pump creates that sodium gradient that allows all those nutrients to pass up, okay, via all these sympads and so forth. And then, of course, you gotta know autoregulation, okay, which is how the kidney accounts for fluctuations in blood pressure, okay, and how you have the myogenic part, which is the contraction of smooth muscle in the blood vessels, or the glomerular rate from the juxtaglomerular apparatus, which gauges the flow rate. So know about that, okay? And then, of course, sympathetic stimulation kind of slows down blood flow through the kidney. Why? Because it constricts the blood vessels, the afferent arteriole. Remember, the sympathetic normally kind of sort of slows down all these other activities. And hormonal regulation, I want you to know antidiuretic hormone, what it does. Antidiuretic hormone is involved in osmolality, okay? For these hormones, I really want you to look at my summary, okay? Because that's what I'm going to ask in the exam, okay? And it increases permeability to water, okay? And it deals with osmolarity, which, because the water dilutes the ions. And then the renin-angiotensin system regulates water content, okay? I know it's kind of sort of counterintuitive when you think the antidiuretic hormone doesn't deal with water content, it deals with osmolarity, okay? So this one, the end result is that, renin produced by the juxtaglomerular apparatus, and then takes angiotensinogen, which is produced by the liver, and then it turns into angiotensin, and then angiotensin 1 is turned into angiotensin 2 by angiotensin converting enzyme, which is called ACE for short, that's produced by the lungs, okay? And then you form aldosterone, which is a mineralocorticoid from the adrenal cortex that will cause more sodium retention, okay? And then because of that, water is also retained. You retain more water. Pregnant women release a bunch of aldosterone so that they retain more water. So that's why they tend to get edema most of the time when you're pregnant, okay? Because of that. All right, so gotta know that. And the atrial natriuretic peptide, the three effects that it does. It inhibits sodium absorption, okay, so that you can lose water because it's only released with high blood pressure and stretching of the right atrium. And then it inhibits ADH because ADH will cause more retention of water. And then it also dilates veins and arteries to relieve the pressure. Okay, so if I might ask about the function of atrial natriuretic, and this is a good summary of it. So look at this, please, okay? And that's it. Sorry we took long, but I'll give you a few minutes for the lab, okay, 10 minutes. So we'll meet at 10 past 3000 B. Thank you guys, I appreciate it.